So now I'm going to start with the structure of the nucleotide. Okay, how the nucleotide structure is formed. Basically, we know that in case of the nucleotide structure, we have three parts or three components. One is the ribose sugar, another one is the nitrogenous base, heterocyclic base, and another one is the phosphoric acid. So how are they organized? How is one nucleotide formed with another nucleotide? How the linkage is being formed? Basically, we are going to see that in this diagram. So first, let me put the heading, the structure of nucleotide. Okay, structure of nucleotide, and in this discussion or in this depiction I am going to discuss that how the its nucleotide structure is formed and how to understand that the entire DNA, how the entire DNA structure is formed with the basic constituents. So we know that firstly there are, as I told, that there are three components of the nucleotide. If I split all those components, then the nucleotide forms with the one pentose sugar. Okay, let me write here only. Okay, it will be easy. One phosphoric acid, okay, one pentose, one ribose sugar, sorry, ribose sugar, 5 carbon sugar actually, ribose sugar, and one phosphoric acid, one ribose sugar, and one nitrogenous base. Now this ribose sugar and the nitrogenous base, the ribose sugar sometimes might be called as a pentose sugar also because it is 5 carbon sugar, so we can call it a pentose. But in case of the depiction of the nucleic acid and all, we have to write it as ribose sugar because ribose sugar is the only sugar that comes from the pentose phosphate pathway. The 5 carbon sugar, that's why it is also known as the pentose sugar. The pentose phosphate pathway gives rise to the ribose sugar, which comes and joins with all these other components. Okay, this ribose sugar and the nitrogenous base is known as nucleoside. Nucleoside. Okay, this is what it is. Now let me draw the diagram. We know that a ribose sugar, in the anomeric ring-like structure, a ribose sugar looks like this. Okay, this is what a ribose sugar looks like in the anomeric ring-like structure. This is what a ribose sugar looks like. And here the aldehyde portion is present over here, the aldehyde group, which is being ultimately linked with the amino acid base. Let me write first the nitrogenous base, sorry. Let me write it as nitrogenous base. Nitrogenous base, okay. This is what the nitrogen. Now here we have, let me write all the other atoms or the radicals present. Here we have the OH, here H, here again we have OH, here H, here also again we have, here instead of OH we have the H part, and this OH of the C, the fourth carbon is going over here, and the fifth carbon is present here as it is CH2OH. Okay. Now how does the nucleotide, this is one ribose sugar along with the nitrogenous base. So this is known as the nucleoside. Now how is this nucleoside forming a bond with another nucleotide, another nucleoside ring? Firstly, when the phosphate, this CH2OH is present right over here, right? Now when the phosphoric acid comes and joins over this particular place, phosphoric acid, which is given, depicted in the form of like this, OH, OH, OH and O. Okay, so this is what the phosphoric acid H3PO4 structure is. This H3PO4 structure will join with this fifth carbon. This O oxygen will join with the fifth carbon along with the formation of removal of the H2O, along with the formation of carbon and oxygen bond. Now how is this formed here anyway? This hydrogen and this OH here is being liberated or dropped off as H2O. Okay, it is being removed, and after removal of this H and OH, the bond that is formed here comes like this. The bond formation happens like this. This is what the bond formation is happening. And similarly, this OH will form the bond with the proceeding proceeding nucleoside or the nucleoside that is present ahead of this phosphate towards the 5' direction. Let me name the carbons first, then it will be easier to understand. This is the first carbon, okay, first, this is second, this is third, this is fourth, and this is fifth carbon. Now this, similarly, this OH of this phosphate or phosphoric acid linked with the phosphorus atom will form a bond along with the same liberation or removal of the H2O with the preceding nucleoside. So here also again I draw another nucleoside. This is what it is. Here also H, nitrogenous base, OH, H, here also H, OH, CH2OH and H. So if I name the carbons, in this case also, it will be same as the previous one, 1, 2, 3, 4 and 5, all these 5 carbons. Now here again the same thing happens, this OH and this H is being dropped off as H2, removed as H2. Now when it is removed, again when it is removed as the H2O molecule, again this carbon will form bond with the oxygen. Okay. And now as the oxygen, the hydrogen bonds are removed, okay, in this case, what happens? This hydrogen is also liberated and this one becomes negative. And the double bond of the, there is no lone pair of oxygen present here, lone pair of electrons or an unpaired electron present here. So that's why this will form, this oxygen atom is sharing 2 electrons with this phosphate present centrally. So there is no unpaired electrons present, okay. Now here, again no unpaired electrons will be present because one electron is being shared with the oxygen. The electron of one electron of carbon is shared with oxygen, and the hydrogen that was present was removed in the form of H2. But in this case what happens, in the course of nucleotide formation, somewhere the depiction remains as in the form of OH. In the form of OH also it remains, or somewhere if the H is not given, it is represented in the form of O minus, because here the unpaired electrons are present. Here oxygen, one electron is being shared with the phosphate and another electron is being shared with the carbon. Here also in this oxygen, one electron is shared with the carbon and another electron is shared with the phosphate. Okay. So this is what, this end is called the 3' end, because this is linked with the third carbon. This is known as the tail of the DNA structure or the nucleotide, and this one 5', 5/5 carbon, this is the 5' head, where again another phosphate group is present. So this part is known as the 5' or known as the head. So this phosphate, similarly in this way, another phosphate molecule will be joined with the carbon with the 5th carbon of this preceding nucleotide, preceding ribose sugar. Okay, and here the nitrogenous bases are mainly the purine bases, purine nitrogenous base, which are mainly the adenine and guanine, adenine or guanine, and the pyrimidine nitrogenous base are cytosine, thymine or uracil. Uracil is present in case of the DNA molecule. Now, this structure that I have drawn here is actually a ribose nucleotide. Okay. Now, if I draw the deoxyribose nucleotide represents the DNA structure, right? DNA means the deoxyribose nucleotide. But so this structure is in case of the RNA. Okay. But in case of DNA, what happens? If I consider this part as the DNA, in that case what will happen? This deoxy, here in the second carbon only hydrogen would be present, oxygen would be not present. Oxygen would not be there, only the hydrogen would be there. So this happens in case of the DNA, and this nucleotide will also not have the oxygen, this will have the hydrogen, and this will continue throughout the entire chain of the nucleotide, polynucleotide strand. And in case of the RNA, the previous structure was being complemented. Okay, it was the OH. Deoxy means the oxygen is removed from the second carbon. From this particular carbon here present, the OH radical is present, the oxygen is removed. That's why it is named as the deoxy ribose nucleotide along with the phosphate. Now this uracil is particularly in case of RNA. Okay, rest all of these are in case of the DNA. Okay, now how is the DNA is double stranded, right? Two strands are present which is running anti-parallel with each other in the DNA structure. Means this 3' tail and the 5' gate is reversed in this direction. Okay, and it is parallel to each other. The distance between the two strands are maintained constant, but this direction is opposite. Here it would be 5' in place of 3' here in the opposite strand, and here it would be 3' in place of the 5'. Now always remember that this adenine base, adenine nucleotide is complementary to the adenine nucleotide is complementary with the thymine. Thymine in case of the DNA, it is complementary with the thymine, or uracil in case of the RNA. Okay, in case of RNA, the uracil is present and thymine stays in the DNA structure. Okay, now so let us now write the uracil part here, because we are considering the double stranded structure of DNA only. The adenine is complemented with thymine, and the cytosine is complementary with the guanine. Okay. Now this adenine nitrogenous base forms two hydrogen bonds with the thymine structure. Here in this bond formation there are two hydrogen bonds, H bonding, okay, H bond. And in the cytosine and guanine there are three bonds, three H bonds. So adenine is forming two bonds with the thymine and cytosine is forming three hydrogen bonds with the guanine. And now always remember the adenine nitrogenous base in case of purine nucleoside, in case of purine nucleoside, if I consider the purine base, nucleoside having purine base, nucleoside having purine base, that is adenine or guanine. In this particular case it would be always the first carbon of the ribose sugar, first carbon of ribose sugar joins with the ninth position. Okay, the ninth position, the nitrogen that is present in the ninth position of heterocyclic ring, heterocyclic base. Heterocyclic base means the nitrogenous base. Okay. So this is forming the first the first carbon is joined with the ninth the nitrogen that is present in the ninth position of the nitrogenous base. This is what it is. And in case of the pyrimidine, if we consider, in case of the pyrimidine nucleotides, if we consider the same thing, then it would be first nitrogen, the nucleoside having pyrimidine base. Here what happens? The first carbon of ribose sugar bonds with the nitrogen present at first carbon of N1, or nitrogen present at the first carbon of the heterocyclic base. Okay, so this is how the bond formation is happening in case of the pyrimidine as well as the purine nucleotides.